Hi, everyone. It's really a pleasure to see you all this morning. So you're here to see 15 young researchers pitching their PhD in 3 minutes. It's really a challenge for them. So please cheer them up, write some comments in the chat. It is an amazing work that they've done. So who are they? They are part of an innovative training network that is funded by the European Union Horizon 2020, and it's called PIPgen. Okay, so you can get more information at pipgen.eu. They are 15. They are really ready to pitch. They've worked a lot to prepare their research in only 3 minutes. And we're going to start really a challenge, and I hope you're going to be happy with that. So we're going to start with Anna Shea Anderson. Let me find Anna. Hello, Anna.
Hi.
Your PhD is called Improving Recognition of Patients with PHTS Phenotypes by easy-to-apply sets of clinical criteria.
Yeah, that is the whole thing.
Are you ready to pitch?
Yes, of course.
Perfect. Then I'll let you do.
Okay, so can anyone tell me what this picture on the left is? Let's be honest, it's a mess. That's exactly what it is. This is also a lot like what it's like trying to find patients with PTEN hamartoma tumor syndrome. This is a rare genetic syndrome, and these patients have a high risk of developing different cancers. And on top of this, they also develop a wide range of non-cancerous tumors. And some patients may also have big heads or autism-like behavior. And with this long list of symptoms, you would think it would be a bit more like this closet on the right, but in reality, it's a lot more like this mess on the left. Just imagine trying to find that one shirt you wanted to wear in the middle of this. Yeah, it's gonna be difficult. You might find another shirt though, which is similar, but it doesn't fit quite right, so it doesn't feel right. And this is another issue we have when trying to find patients, because a lot of their symptoms are also found in other patient groups. So different patients may look similar, and because of this, our patients aren't being found. So it's clear that we need to sort of clean up this mess, but how do we decide what to wear and when to wear it? This is what I'm trying to figure out. What should remain in the closet of symptoms? There's definitely a few cancers that will stay on the shelves, but maybe a few of the others can be put away in storage or taken somewhere else. And what do we do about big heads or non-cancerous tumors? Maybe some of them can be ignored if they aren't as relevant as we first thought. There's definitely a few things in here that do not belong, but we don't actually know that until we actually start organizing and cleaning up the mess. So organizing everything will make it easier for us to match and combine different symptoms and hopefully find patients, because they need very specialized care. And organizing everything will also help others to find them, because they will then have a very clear overview of what they need to look for. And obviously, knowing where to look and what to look for will help us, but most importantly, it will help the patients, because finding these patients at the right time is instrumental in preventing cancers from developing in the first place. So, to summarize, sometimes less is more, or at least it's easier to organize. Thank you.
Well done, Anna. Thank you very much.
Thank you.
Well done, and thank you for being the first. It's never easy to start, so you did very well. Thank you, thank you, thank you. We've put all the links actually in the chat, so if you want to connect with Anna, don't hesitate. You have LinkedIn, Twitter, and some other. And I'd be now Devika's turn. So thank you and bye. And now Devika Kurup with the title of PhD, role of PTEN, PI, mutation in increased risks for autism spectrum disorder.
Yes.
Right? You're going to explain
Yes, yeah.
that in a way simpler term than the title. Are you ready?
Yes, I'll try my max.
Okay, so I leave you. Do it. You have your 3 minutes.
Thank you very much. Hello, everyone. Like you heard, today I want to talk about mutations. But don't worry, I'm not going to bore you with technical details. Instead, let's think of something familiar, like baking a cake. Have you ever accidentally skipped a step or misread an instruction in a recipe and ended up with a cake that didn't quite taste right? Maybe you burned a cake. We all have at some point, right? But just like how you need a recipe to prepare a cake, each part of your cell needs specific instructions that we call genes. Now, these are present everywhere in the body, like we see in this person with yellow dots representing normal genes. And just like how some of us like to experiment with cake recipes, our genes can also go through some changes, which we call mutations. Imagine these dark dots on the people in pink and blue as an effect of one of this mutation, but we'll get to that soon. Before that, it is more important to let you know that mutations can be good, like discovering a delicious cake recipe. But they can also be not so good, like when the cake turns out to be half baked. When our genes go through mutations, it can affect how our body works, and sometimes it can cause serious problems. Now, let's take a closer look at the person in pink who has this specific mutation that I am interested in. As we see here, people with this mutation seem to be too weak to fight off infections. This made scientists wonder if this mutation affected the immune system, and indeed, that was the case. Because this mutation clearly affected immunity, for a long time, scientists mainly focused on the immune system of these patients. However, more recently, doctors started noticing something unusual about the patient's behavior. They have trouble coordinating their movements and performing simple tasks, and so on. This led to an important question. What does this mutation do to the brain? This is where my PhD comes in. We at Erasmus MC want to understand whether this mutation affects the brain, and if so, how it affects behavior. By doing so, we can gain insights into why these patients behave the way they do and hopefully develop new ways to help and treat them. But that's not all. Studying mutation in the brain can also help us understand how the brain works in general, which has broad implications for neuroscience. It can also provide insights into certain brain disorders like autism spectrum disorder, leading to the development of new diagnostic tools and strategies that could improve many lives. So, let us continue to unlock the mysteries of our genetic recipe and work towards a better future. Thank you very much for your attention.
Well done, Devika.
Yeah. Thank you.
Thank you, thank you, thank you. And how did you feel during the pitch?
It was really daunting, but I must say it was a really a very productive output, which I think I made. And it's nicer to explain to people who really don't understand the technicalities, you know. So it was a really good experience.
Yes.
Very happy we did this. Yes.
Well done.
Yeah.
Well done. And again, we've put your links if people want to talk with you in the chat. So if anyone wants to join, can. Thank you,
Please feel
Devika.
free.
Well done. And now we're gonna and we're gonna go to the next one now. Thank you.
Bye-bye.
So now it's the turn of Emanuele Maya Nola, understanding vascular malformation in PhDs. Hi, Emanuele.
Hi, everybody. Hi, Elodie.
Are you ready to pitch?
Yes.
Okay, so I'm going to leave you for your 3 minutes.
Okay, so do you prefer to drive a car in a street that looks like the left image or the right image? I guess the answer will be the right image. Now, instead of thinking about streets, imagine that these are the structure of blood vessels. Blood vessels are the streets, the highways that transport our blood throughout the body. Blood is fundamental because it carries the nutrients and the oxygen that are essential for our life. So, try to visualize if our blood vessels have a completely disorganized structure as the image in the left. We call this condition vascular malformation, and it means there won't be a correct distribution of the blood in our body, and depending in which part of the body is found, could represent different kind of problems that could vary from a little bit of pain to an amputation of the limb. So my aim is to understand why these vascular malformations appears in the body and how we can repair them in order to pass from the image from the left to the image of the right. First of all, the blood vessels are composed by many little units that are called cells that collaborate all together to give a perfect shape to each blood vessel. But how they can collaborate in such a perfect way? Thanks to the instructions that they have inside themselves that are called genes. These instructions are read and assembled by the cells, producing little components called protein that give the shape to every single cell. The problem is when these instructions are written in a wrong way. So, in this case, it means that a gene is mutated, so we have a gene mutation, and the proteins produced by the cells are irregular, and these can lead to a vascular malformation. So now the challenge is to find these instructions that are misspelled and try to fix them. So I'm studying a group of people that present these vascular malformations, and thanks to some computer programs, I'll be able to identify these new genetic mutations. How? It's like to have many different pieces of a puzzle that is composed by 100,000 of pieces in which some of them are not belonging to the puzzle that I want to assemble. Thanks to the help of the computer program, I'll be able to solve in a correct way the puzzle and find the new genetic mutations that cause the vascular malformation. Once identifying these new genetic mutations, I can talk and suggest to the doctors which is the medicine that could repair the misspelled instruction. So it will be repaired the disorganized structure of blood vessels in order to be sure that your blood never have to drive in the streets of the left picture.
Thank you, Emanuele.
Thank you.
Amazing. I think now we're going to think of you when we're stuck in traffic, or we are going to have to take a very, very complicated road. We're going to think of you and the blood cells and your work. Amazing. Thank you very much.
Thank you.
Again, the links to join you are in the chat. So if anyone wants to talk to you, they join you on LinkedIn or whatever, and they can ask you more questions about blood vessels and traffic. Well done for the pitching. Thank you very much, Emanuele.
Thank you.
So now we're going to go to the next one, Fateme Omidi. And yes, she's here. Hello.
Hi, everyone. Hi,
So your title is Improve Accuracy and Time to Diagnosis of Rare Disease by Developing AI-Based Algorithm.
Yes,
Are you ready to pitch in 3 minutes?
I am.
The screen is yours.
Okay, well, today I have a challenge for you. Look at these paragraphs and find as many as misspelled words and errors as you can. You have 5 seconds to do that. Ready? Go. And time's up. How many did you find? There are actually 3 of them. Humans in the first line, each in the second line, and the first sentence of the second paragraph is despite our differences, not our references. As you can see, differences makes much more sense here. Now, if you think it was kind of hard to do that, I want you to think of finding errors in a book with 10 million pages. That book is our genome. We can think of our genome as a book with 10 million pages, and 3.2 billion letters. Like books, there are errors in our genome, which we call them variants. On average, there are 3 to 4 million variants in a genome. Now, take another look into these errors. Two of them you can easily read and understand the meaning, and one of them changes the meaning of the whole sentence. Same thing happens in our genome. In a healthy person, those 3 to 4 million variants, they are not disease-causing. They don't affect the way a body works. However, in a case of a patient or a research study, we are looking for those rare variants that can change the meaning. We can refer to them as target variants or disease-causing variants. There are two main challenges in order to find and identify the target variants. First, it is time-consuming because an expert needs to study huge amounts of variants and decides if they are disease-causing, if they are our target variants or not. And second, sometimes we might miss a target variant for some cases because of different reasons, for example, technical or knowledge issues. So, my project is to address these two challenges. First, we are going to design a strategy mainly based on artificial intelligence, in which we will teach the computer, the machine, how to decide whether a variant is disease-causing or is not. And with this, we will decrease the time. And second, we will provide different layers of information for each variant. So within our model, we can study the variants from different aspects. And with that, we will increase the probability of finding the right variants. So in the end, with this workflow, we are able to find our target variants faster, and we can find them for a higher number of cases. Thank you for listening. That's it for me.
Well done, Amy. Thank you. Thank you. So how was it preparing the pitch?
Well, it was really challenging because the time was limited and also we wanted to make it really understanding. But I really like that.
Okay, it was amazing. Well done. We have Jean-Mehdi Grandjean, that is the little fairy of this live, is pasting all the links to join you on social network. I hope there is no misspelling and we can actually join you all, but normally it's all good. Yeah, trying to make a link with your talk, but I'm not sure it worked well. So don't hesitate to contact Fatime, and thank you. We're gonna go to the next one now. Well done. So the next one, where is she? Is Ilaya Galasso. Hi, Ilaya.
Hello, dear. Hi, everyone.
So the title of your PhD is Understanding the Physiopathology of Growth.
Yes.
Okay. Are you ready to pitch?
Yes.
Okay, the floor is yours. You have 3 minutes.
So I work on a disease that affects only 14 people in 1 million. Have you ever heard of PIK3-related overgrowth syndromes? Until 2 years ago, I had never heard of them, and the reason is because they encompass a group of very rare and difficult to classify diseases. Patients affected by this disease present abnormal growth of various body parts or defect in the vascular system, and this is due to changes known as mutation in the PIK3CA gene. This gene is involved in making a very important protein that is actually fundamental for the cells and help regulate their growth, their division, and their survival. You can imagine how important it is for the cells having this protein working properly, and the problem arising when it's not well regulated. Since PIK3CA mutations are not inherited but originate during the embryonic development, they are not present in all the cells of the body. Imagine the cells that compose the early embryo as the tiles that compose a colorful mosaic, with each of them having a different color. Now imagine that at a certain point, one of them acquires a mutation that makes it lose its color. All the cells deriving by this one will be white. In the same way, only the cells and consequently the tissue deriving by this one will carry the PIK3CA mutation. Depending on the time in which the mutation occurs and the cell type, different tissue may be involved, individually or in combination, and have different clinical consequences. We in the lab noticed that cells affected by this PIK3CA mutation show a different way of getting nutrients in comparison to their healthy sisters. In fact, they have a predilection for glucose that distinguish them from the others. So now we have two objectives. The first one is to explain how this change happened in the first place. And the second one is to exploit this particular characteristic of the mutated cells to target them with a specific drug. In the lab, we have already found a drug to treat the patient, which has now become the gold standard for the treatment. But there is still room for improvement. What we are planning to do now is to combine the drug with other treatments specifically directed to the mutated cells. Some of you may be wondering, how will it be possible to target treatment only against mutated cells? Well, probably thanks to their specific predilection for glucose. The idea is to try to make glucose less available or more difficult to use for these cells, and thus to try to make them more susceptible to the therapy. Doing this, we hope to develop a target therapy and improving the quality of life of those patients that are less responsive to our gold standard treatment. Thank you.
Well done, Ilaria.
Thank you.
Thank you for your 3 minutes. You felt you were a bit stressed? No, you were.
I can't hear
Ilaria?
you. Yes.
Oh, sorry, I was asking if you were a bit stressed about the pitch.
Yes, just at the beginning, but then it went well.
Yes, it went really well. Thank you for explaining mosaicism in some disease. If you want to know more and to contact Ilaria, the links are in the chat. Thank you, Ilaria. We're going to go on the next one now. So we're going to welcome Janki Bat. Hello. Oh, no, actually, sorry, Janki. We're going to actually welcome Ilaria Rondon. Hi.
Hi.
Sorry. So Ivana Rondon, sorry, I completely, my list was a bit, I missed the list. So hi, Ivana.
Hi, Elodie.
Your PhD is called Polygenic Alterations which emerge from monogenic PTEN loss-driven prostate cancer.
Yes.
And you're going to make it simple for us in 3 minutes. Are you ready to pitch?
Sure.
Okay, you have your 3 minutes.
Okay, so you have probably heard about prostate cancer, but have you ever thought about what are the exact conditions for his creation? And this is a very important question that researchers need to know to understand this disease. And it has served as an inspiration for my PhD project. So for you to understand this, I will try to introduce you a kind of recipe for creating prostate cancer. So the first ingredient, and highly important for all types of cancers, are errors that happen at the DNA level that are known as mutations. And these errors cause that your cells stop working properly. The second ingredient is aging, and this is based on the observation that prostate cancer is a disease that is observed in men when they age. The third ingredient is caused by lifestyle factors that like having a very bad diet, not doing enough exercise, and having too much stress. And all these ingredients combined are essential for the maintenance and growth of prostatic tumors. However, when you have the tumor already created, it makes it difficult to understand what was his cause and to try to study his development. This is like, for example, when you have a cake and you want to know what are the exact ingredients that were added to the cake and in which proportions. So to overcome this issue, the researchers use mouse models to try to add some of the ingredients to induce a prostate cancer in the mice, and to have a model that approached as close as possible to the human one, but with the advantage that it can be studied in a controlled way. And the interesting question about this is how the information from the analysis in mouse models can be translated into the humans, and how this approach can reveal strategies to fight and predict aggressiveness of this disease that is highly frequent in all men around the world. Well, this is the aim of my PhD project, and these are the kind of questions that I'm trying to solve. And to do so, I will try to use computational tools to try to reveal what kind of mutations, or what if there are a specific mutation that happen in prostate cells that are associated with the aggressiveness of this cancer. So thank you very much.
Thank you, Ivana, for making everything really, really clear. So again, the links are in the chat, so if we want to contact you, we can. Well done for pitching. I hope it was fine and not too stressful. It's really a big challenge for all to in minutes. I think you made it really simple.
Thank you.
Oh, sorry, we lost Ivana, so we're gonna go to the next one now. Junkie Bat. Hello, Junkie. Sorry, I made you have a little heart attack just before having you called a bit too early.
Yeah, it's okay. I wasn't expecting that.
So your PhD title is to screen and analyze the drug-like profile of profound targeting PI3K, right?
Yes. Yep.
Okay, so you have 3 minutes to explain it to us.
Thank you. What if I told you there's a bunch of grapes inside your lungs? Weird, right? But it's true. Your lungs contain air sacs that are actually shaped like grapes, which help you breathe better, which help you breathe. But how can grapes fit in your lungs? To put the size in perspective, imagine how many grapes can fit in your mouth. 5, maybe 10 if you have a big mouth. Now imagine how many ants can fit in your mouth. I said ants. Don't ask me how, but the answer is 248. Well, these grape-shaped air sacs are actually 20 times smaller than the size of an ant, which is why there's 300 million of these inside your lungs. But just like grapes in a vineyard can get infected with pests and insects, similarly, a few million people in the world can develop a disease that causes damage to their air sacs, making it difficult for them to breathe, causing coughing, eventually lung failure, and even death. So how do we solve both these problems of the grapes and the air sacs? So for grapes, if they're infected with pests and bugs, you can use pesticides. For humans, I wouldn't recommend pesticides. Well, luckily for us, there are drugs that can actually stop this lung damage from progressing and causing any problems further. But just like pesticides can successfully stop the pests and insects from damaging all your grapes, these drugs can also stop lung damage to your air sacs. However, similar to the pesticides, you know how they can cause environment pollution or toxicity to human health. These drugs can have additional effects such as damage to healthy body parts like your liver. This is why my PhD project focuses on developing a drug that not only stops the lung damage from progressing, but it also does not have an effect on healthy body parts. So no side effects. Think of it as an eco-friendly pesticide. Yep. Thank you.
Thank you, Janky. I won't ask how you know about how many ants you can fit in your mouth.
I Googled it.
You Googled it. Okay. Okay, I'm feeling better now. I was thinking, did she actually try? Good. Well done for pitching. Thank you very, very much for that. And we're going to welcome the next one. 15. Yeah, it's quite many. So thank you, Janky. And now it's the turn of Alberto Collado. Hi.
Hi.
How are you? You feel ready to pitch?
I am, yeah.
Okay,
Ready to
so
go.
we're going to listen to you for 3 minutes. Yes.
So, imagine this. Your alarm clock goes off, you wake up, look at the clock, 7:30 in the morning, and you remember, today is the day. Today is the day for the interview that you have been preparing for so long. And you go to the shower, and the water temperature is just right. And then you make yourself some breakfast, and the toast is toasted beautifully. And while you're getting into the car to go to the interview, you think to yourself, you know what? Today's going to be a good day. And as soon as you hit the highway, bam, traffic jam. You can't see the end of it. There seems to be some damage in the road ahead. And now it's not only your day that has been ruined, but for many other people in the traffic jam. And in that traffic jam, you have baristas that were going to be serving coffees for workers to wake them up. And you have clerks and administrators that were going to be making sure that their offices run in a timely manner. And you have delivery drivers that were going to be giving out parcels. What I'm trying to say here is that the road communicates different parts of society, and damage to roads means that there is damage to society and how it communicates and how it works. This is very similar to how our blood vessels work, because believe it or not, our blood vessels are basically highways in our body, and they serve to deliver not only microscopic workers, but also oxygen and nutrients, which are essential for the proper working of our body. So where there is damage to a specific part of our body, there is an issue with delivering those oxygen and nutrient to that area. So when there is a wound or an injury, and the first step to be able to heal that wound or injury is the regrowth of these blood vessels. So the repair of the road for them to be able to deliver again oxygen and nutrients. This is where my PhD comes in, because our lab has developed a new drug that acts very good on blood vessels by accelerating their growth and thus accelerating the regeneration and the repair of wounds and injuries. We could think of this as, road repair typically takes a long time, right? And you can think of this like people shoveling away using their bare hands to try and repair the road. Our drug, what it does is give them the tools that they need to repair it faster and more efficiently. So we suddenly give the workers bulldozers and excavators and all of this fancy equipment. Now suddenly, instead of shoveling, they can just work their way around and repair the road, and instead of months, it's going to take weeks or maybe even days. We think our drug is going to be very beneficial to translate this into the clinic and the management of complicated to cure injuries and wounds and so on. Thank you.
Well done, Alberto. Thank you for your amazing pitch. Again, it's a bit like Emanuele. We're going to think of you next time we're stuck in traffic jams and think about your research.
Yeah, sorry about that. Can I give a
bit when
of
we drink
PTSD?
Coffee, actually, because or drink coffee, because we talked about the barista that save our lives and caffeinate us. Thank you very much. Again, the links are in the chat if people want to join you and to contact you. And yeah, we have someone that, you have a little fan club in the chat. Amazing.
Yeah.
But actually, you were one in the middle, so we're halfway through all the pitches. So we're going to continue. And actually, lots of people are on YouTube. It's very nice to see you all. And yes, thank you, Alberto. It's very nice to see so many people on YouTube. You can hi to people watching it on replay as well. So now it's the turn of Leki Tang. Hi, Leki.
Hiya. Lovely to be here.
You're going to talk about using cells exhaustion in a model of APDS, right?
Yeah.
And it's going to be all clear for us in 3 minutes. Are you ready to pitch?
Yeah, I'm ready to go.
Okay, I'm leaving you, and this is your 3 minutes.
Cool. Well, have you ever wondered why cancer cells tend to spread to certain parts of the body more than the others? It's because of something called the seed and soil theory. Just like plants need the right environment to grow, cancer cells need the right environment to thrive and spread. This theory helps explain why certain types of cancer are more likely to spread to certain organs. Think of it like this. Cancer cells are like seeds, and the organs in the bodies are like soil. Just as some seeds grow better in certain types of soil, some cancer cells are better suited to grow in certain organs. This is because different organs have different environments, like different levels of oxygen or nutrients that affect how well cancer cells can grow and spread. Scientists are trying to find ways to make the environment less hospitable for cancer cells. While not only targeting the cancer cells themselves, we focus on preventing them from spreading and growing by employing our own immune system. In our group, one protein called PI3K delta has been of our long-standing interest, as it involves in the activation, differentiation, and effector function of multiple immune cells. PI3K delta has been successfully targeted in the treatment of lymphomas, a type of liquid tumor that affects the lymphatic system. However, its potential role in the treatment of solid tumor still remains unknown, particularly to treat solid tumor that could spread throughout the body, and that's been the focus of my current study. To test this, we injected mice with cancer cells to stimulate lung metastasis, a condition where skin cancer cells can spread to the lung. Strikingly, we found that the mice with a hyperactive version of this protein had a reduced tumor growth in the lungs. What's exciting about this is that this observation suggests targeting the PI3K delta pathway could be useful in treating solid tumor by enhancing the function of our own immune system. This could be particularly important for cancer immunotherapy, where transferring immune cells into our own body has been a promising strategy for cancer treatment. Overall, the seed and soil theory and our research on PI3K delta are just a few examples of how scientists are working hard to understand and fight cancer. While there is still much to learn and do, every step forward brings us closer to better treatments that can save lives and improve quality of life for patients. Thank you very much.
Thank you, Leki. Thank you. Well done.
Thank you.
So we're going to go to the next one. We still have a few to go through, so thank you. We're going to welcome now Louis Maes. Hello, Louis.
Hello, can you hear me?
Yes. Your
right.
PhD is called, so your PhD title is to identify PI3K vascular-related therapies which enhance immunoregulation flux, right?
Yes, more or less, exactly.
You're going to make it all clear in 3 minutes for us.
I will try.
Are you ready to pitch?
Yes.
Okay, so this is your 3 minutes. You can go now.
All right. So I think it's safe to say that a lot of you know what IKEA is. Honestly, it's great. It's a perfect Saturday afternoon, right? You go there, you eat the meatballs, and probably you will take home a piece of furniture. For example, this Billy closet. Now, if you come home, you already have all the building blocks to make the closet, but how do you actually assemble it? Probably you would refer to the manual. In the manual, it contains all the instructions that you need to put all the pieces together in the right order that you will get a perfectly working closet, right? Well, your cells of your body do more or less a similar thing. So when they need some furniture, they would also refer to their manual, which is the DNA. So the DNA is basically a huge collection of all of the instructions that all of the cells in your body might need. For example, and then of course the furniture of your cells will determine its function. For example, the muscle cells have the furniture to contract, or the fat cells have the furniture to store energy in your body that it might need later. Now, the furniture of your cells we refer to as proteins. So proteins are the real molecular machines that perform the functions that your cells might need. But what if something goes wrong with the instructions? Imagine that there is a printing mistake in the manual of IKEA. Probably you will assemble the closet wrong now, and you will end up with some very funny looking closets that cannot perform their function at all. So it's more or less the same with DNA. If there is a mistake in the DNA, you will end up with a protein that cannot perform its function well. And a mistake in the DNA is called a mutation. So I'm studying the type of mutations that cause the protein to work a little bit too well. Normally, this protein is involved in the survival and the growth and the multiplication of the cells. But with the certain mutations, it will become too active and the cells will start to grow and to multiply too much. I'm studying this specifically in the blood vessels. When the blood vessels have these kinds of mutations, they start to overgrow like a jungle in your body. This can, of course, manifest in different ways. So sometimes it's very small that you will not even notice it, but other times it will be very problematic and you'll get a huge head or a huge leg, and this can, of course, be a problem. There's an example of a little girl that came to the hospital and she was paralyzed from the waist down because her blood vessels were pressing against her spinal cord. Now, luckily, they were able to treat her by using a drug that can inhibit this super active protein, and now she's able to do all the normal things that a little girl should be able to do. So how am I doing this? I am looking at mice and their blood vessels that carry these kinds of mutations to better understand the mechanisms. But we also have a way to isolate the blood vessel cells from these kinds of patients and study them in a dish. So finally, my research will help to understand the mechanisms of how these mutations can cause overgrowth in the blood vessels and, of course, how we can treat it.
Thank you, Louis. Well done.
Thank you.
So I like that with this live, we're going to think about you know, the whole PIPgen group when we drive, when we drink coffee, and now when we are assembling IKEA furniture.
Exactly. I was driving past IKEA this morning and I was like, I'm going to present about you today.
Well done. Thank you very much. I think we all had the problem of trying to fix a furniture. So thank you. Thank you very much for your great. Thank you, Louis.
Thank you.
Again,
Bye-bye.
the links are in the chat if you want to reach him. Bye-bye, Louis. So we're going to go now and see and welcome Maya Shauki. Hi, Maya.
Hello.
So your PhD is about drug evolution in B-cell lymphoma patient under idealism, right?
Idealism,
okay,
exactly.
You're going to make that all clear for us in the next 3 minutes. Are you ready to pitch?
Yes, I am.
Okay. You can go.
So imagine that you break up with your partner and to console yourself, you start eating Nutella. First 1 spoon and you feel better, but after you start eating more. So 2 spoons, then 3, until you finish the jar and you find yourself stuck in the bathroom with crazy crumbs. So Nutella is so tasty, right? I can eat a spoon and it gives me a feeling of well-being and happiness. But if I eat too much, it makes me sick. In other words, this can happen also with drugs. To be clear, taking drugs to treat cancer is important and we should not stop taking it. So drugs are necessary to treat cancer, but for some patients can also create toxicity in a long term with different side effects such diarrhea, colitis, hepatotoxicity, and infection. In particular, in CLL, chronic lymphocyte leukemia, a blood tumor, there is a cell population called B cells that change and grow out of control, attacking certain organs of the body, such tonsils, spleen, liver, causing the death of the patient. Fortunately, drugs have been created to block this tumor and increasing the standard of living patients. Despite the efficacy of these drugs, after around 6 months of treatment, these drugs have been associated with a strong toxicity and major side effect in patients. So basically, long-term treatment of these drugs is impossible for these patients. We therefore wondered whether decreasing the dose could maintain a high efficacy while decreasing the toxicity. The aim of my PhD is to demonstrate the efficacy of low doses. How? So first of all, I want to study the efficacy. So I want to compare the efficacy of different drugs at low doses with an indication of lower toxicity event in cell from patient and in cell line. And after, we want to focus on the toxicity. So we will study some cells called T cells that play a major role in immunity. And this population, after the treatment with these drugs, decrease. So we want to understand why. To conclude, for this tumor, different drugs are efficient, but at the same time can create toxicity, causing also the death of the patient. We want to prove that low doses are very efficient and do not cause toxicity, improving the patient life. Thank you.
Thank you, Maya. Thank you, Maya, for your pitch.
Thank you.
It's much clearer now. Thank you, thank you. If you want to contact Maya, the links are in the chat. Well done, and thank you again. So we're gonna go to the next one. Bye, Maya.
Bye.
So now we're gonna welcome Shanzar.
You can stay.
Hi, Shanzar. You're in the same as Maya, so we have Maya in the background that just pitched.
Yes, that's correct.
So your PhD is called Mechanical Stress and Signal Integration by by PI3Ks for the control of tumor growth.
Exactly.
Are you ready to pitch?
Yes.
You have your 3 minutes.
So imagine a situation that you're being told that you have about 12 months since you have been diagnosed with pancreatic cancer. And sadly, that's the reality for majority of pancreatic cancer patients. Cells in our body are different from each other. Like skin cells have unique characteristics that are missing in nerve cells. And nerve cells, on the other hand, are obviously different from blood cells. And same holds true for different types of cancer. Characteristics and vulnerabilities of one cancer type are not necessarily the same for the other cancer type. Treatments that will work for blood cancer wouldn't be efficient in treating pancreatic cancer, since pancreatic cancer has its own unique characteristics, right? So what are those? There are many, starting with mutations and ending with cell of origin. And one of such characteristics of pancreatic cancer is that it develops a very mechanically stressful environment around itself. So one way to picture it could be an analogy to a tube. When we take metro during normal hours, there is plenty of space and everyone can take a place to sit, right? Same as in normal pancreas. Cells are well and correctly placed in the organ. While if we take metro in rush hour, we got jumped in this restricted space, pushed and pulled by other people against the walls, against each other, very stressful, both mentally and physically. And same happens as pancreatic cancer develops. Cancer cells grow and in this restricted space and create a very mechanically stressful environment. They push and pull against each other. And this leads up to the project of my thesis, where I study how this developing mechanical stress actually affects pancreatic cancer cell behavior. To put it simply, I push cancer cells and I see how they respond. So the first results in this project show that the pancreatic cancer cells respond to this stress, to this mechanical stress, by changing their identity and also by trying to create a cushion around themselves, a layer of protection, I would say, by producing these proteins known as extracellular matrix proteins, or simply put, proteins surrounding cells around. So which you could see at the bottommost picture. The ultimate goal of this project is to understand how this mechanical stress affects pancreatic cancer cells and whether changing this stressful environment could help us to improve current therapy options. Thank you very much.
Thank you, Shanzar.
Thank you.
Thank you, well done. And I think with this live, we're going to think about all your work all the time, because now it's when you're in the tube. So all the analogies are very great. Thank you for explaining to us. And actually, cells can feel stressed because they are pushed. So this is a very big information that we can take from.
Yes.
Thank you, Shanzar.
Exactly. Thank you. Bye.
Well done. Well done with your pitch. And now we're going to welcome the next one, which is Sarah Aljaraseto.
Hello.
Hi, Sarah.
Hi, Elodie.
Your
How are you?
PhD title is good, good, good. I'm so happy to see you all pitching. You know, it's amazing. Thank you for waiting because you're one of the last ones. And your PhD is called The Role of PI3 Delta in Solid Tumors and its Modulation Using Novel Pharmacological Tools.
Very catchy.
Just to warn you, yeah, you're gonna, but I know how you're gonna explain it. Just to warn you, it's a PDF, so we won't see the animation, but it's still gonna be a great slide. Sarah, are you ready for your 3 minutes?
Yes, I am.
Okay, so the screen is yours.
Great, thank you. Right, so did you know that we have many control panels in our bodies? Our bodies consist of various intertwined systems like the nervous system or the circulatory system. And like any busy place, there needs to be a method to keep all the systems under tight control and prevent the signal traffic from going crazy. Our body uses control panels to regulate all the signals from all these systems. And we call these control panels post-translational modifications. Cells normally use protein molecules to communicate with other cells and systems to give a response. The control panels apply different modifications to these proteins to turn them on or off like mini switches that activate or deactivate a circuit. In the control panel of post-translational modifications, 4 main types of switches or modifications can be found. My project focuses on a specific type of switches called phosphorylations, and we want to study the modifications that our bodies make to an important protein called PI3K delta. And this protein goes crazy and becomes overactive in some immune diseases and some cancers. So my project wants to investigate if something went wrong in the control panel that flipped the switch of phosphorylation in the PI3K delta protein and turned it on when it shouldn't be, resulting in disease. And we also want to understand how and why this happens. To achieve this, I use cells from the immune system called B cells, which are rich in the PI3K delta protein, and I apply different conditions to these cells to test for different responses in the phosphorylation switches and see if I can identify a particular switch that is responsible for the overactivation of this protein in disease states. We also want to make a drug that can destroy cells in which the PI3K delta makes it easier to monitor the switches in the protein. This model tests delta protein in our bodies.
Sarah, sorry, I have a bit of a connection issue. You finished, right?
No worries. Yeah, I did.
Sorry. Yeah, I had a bit of connection issues, so I'd rather, wait a bit too long than actually crashing your talk. Well done, and thank you. Thank you very much for your pitch. Well done. And yeah, and if we want to contact you, the links are in the chat again. So well done, Sarah.
Thank you.
Thank you very much. And we're going to go on to the next ones. It's been an hour almost. They see everyone going, and it must be really stressful. Shalmali, how are you?
Hello, Elodie.
Hi.
Hi, Elodie. I'm good. How are you?
Good. I'm super happy to see all your pitches taking life. Your pitch is about biomarkers of chronically, hyperactivated PI3K delta in patients with APDS, right?
Yes.
So many acronyms, you're going to make it all clear for us. It must be difficult to be waiting all the time. I'm not going to make you wait more, and you have your 3 minutes. Are you ready to pitch?
Yes, I am.
Okay, perfect. So I let the screen I let the screen to you.
Okay, so imagine that you have a fever and you go to the doctor to find out what's wrong. The doctor draws some blood and finds that you have an increased number of white blood cells and some proteins. This tells the doctor that you most likely have an infection. And then the doctor can decide on further tests and determine the best course of treatment for you. These two things, which help the doctor diagnose you, are called diagnostic biomarkers, and they can be thought of as a crystal ball for the doctor. If they are there, it means that you have a specific disease. Similarly, you can also get crystal balls that can tell you if a patient will respond to a particular drug. And this is the focus of my PhD. In order to do this, I will first analyze the blood from patients with a damaged immune system that belong to a specific disease group. And from now on, I shall be referring to them collectively as patient A. The crystal ball in this case is a special group of cells that only exist in patient A and not in healthy people. The reason why these cells exist in the first place is because of mutations in two genes that leads to an increased activity of this particular protein, which in turn has drastic effects of changes in the frequencies of various cellular populations of the immune system, and this in turn damages the immune system over time. So, if these cells are present, that means these patients have increased activity of the protein, and therefore these cells are the marker of this increased activity. Once I have found these cells or the crystal ball, I will try to find it in patients of other diseases that belong to the complex disease group. And they have symptoms that are similar to patient A. So it is possible that a small fraction of them might also have increased activity of this protein. And if so, this crystal ball can help us find them, and their symptoms can be reduced with treatment with a drug that is specifically targeted, and its function is to normalize the overactivity of this protein. And with this, their symptoms will reduce, and they will also get much less side effects. This is also the standard treatment for patient A. So in conclusion, the main aim of my PhD project is to extend personalized treatment options to patients of complex diseases where the relationship between the cause of the disease and the best treatment option is often blurred. Furthermore, this can also help doctors efficiently and accurately differentiate between patients that have similar phenotypes or similar symptom presentation by differentiating between them at the molecular and cellular level, as it is the information at this level which often gives the doctor the best indication of the match to be made between the patient and the drug that will most likely help them get better. Thank you.
Well done. Thank you.
Thank you so much.
Thank you for your pitch. Thank you for being one of the last. Well done for your pitch. And I hope you're going to see amazing things during your PhD in your crystal ball, and you're going to manage to
hopefully
improve the future of patients. Thank you very much.
Yes.
And we're going to leave you now to welcome the very last one of today, Shanlin Tong. Hi, Shanlin.
Hello. Hello, Elodie. How are you?
Good.
Great.
How do you feel? You okay? You had to wait quite a bit. How is it to be the last one?
I mean, it's great. I've been watching everybody's pitch, and very exciting, and to see everybody's work.
Pitches.
Absolutely.
Amazing. Yes. So your title is Understanding the Role of Activated PI3K Delta Mutant in B-cell lymphoma, right?
Yes, that's true.
And I'm going to do a little trick because your slide wasn't, so bear with me a second. We're just, this is your slide. Yeah. Are you ready to pitch?
Yes.
Okay, so I let you go. This is, yeah, the screen is yours.
Yes. So imagine sitting down to a delicious Chinese meal and eagerly taking a bite of a new dish, only to find that it's a little bit too spicy for your taste. And you might wonder what makes the dish so spicy. It could be that a particular type of pepper, a secret blend of spices, or something else entirely. And if you love Chinese food just as much as I do, you even go that extra mile to figure out what makes that dish so interesting. And this can be a quite a complex process, just like to study the biology of cancer. It's a complex disease because it can arise from various factors, such as exposure to toxins, genetics, and lifestyle choices. And one of the key features of cancer is its ability to grow and spread uncontrollably, eventually leading to the formation of tumors and metastasis, which can be life-threatening. So to better understand this process, my PhD research is to find the genes that are responsible for cancer development with the help of powerful genetic editing technologies, which basically allow me to manipulate the genetic code of cells with unprecedented precision. So the idea is simple. I'm going to engineer the DNAs of cancer cells, remove specific genes, and just to see how this would affect the behavior of cells. And this approach would allow me to identify which genes are essentials for cancer survival, just like leaving out different ingredients from a recipe to figure out the secret ingredient that makes the dish spicy. And by identifying these key genes, researchers would in the future be able to develop more targeted and precise therapies that can be used to treat cancers and more efficiently, rather than using a one-size-fits-all approach. So doctors can tailor treatments based on the unique cancer profiles of that patient and targeting genes that are responsible for driving the cancer. So this research has the potentials to improve the lives of millions of individuals and families affected by cancer, giving it a better outcome for the patients and hopefully a bright futures for everyone.
Well done, Charlene. It was amazing. Thank you.
Thank you.
Thank you, thank you. So you were the last one. It was great. It was great discovering all the work of all of you. Thank you for making it simpler. And again, I think we're going to review every step of our lives now. You know, coffee, driving, going in the tube, and now whenever we cook some Chinese food or even whatever food, we're going to think about the secret ingredient that is there, and we're going to think of you.
Thank you.
The links again are in the chat, and thank you very much. Thank you very much, and you know, people can stay in touch, and we're gonna leave you now for the closure. No, sorry, for the finishing of the live. Sorry. Bye, Charlene.
Bye bye.
Okay, we're done. We're finished. Thank you very much for following this live. It was amazing. We had 15 PhD students from PIPgen network pitching their PhD amazingly well, making it very simple for us. It was really a challenge. So thank you for following that. I also would like to say thank you to Jean-Mehdi Grandjean that is behind and is making sure that all the links are there, the slides are there and everything. It's amazing to work with you, Jean-Mehdi, so thank you, thank you. And well, you can go to the website pipgen.eu that might appear in the chat as well to know more. That is in the description of that video. So thank you very much everyone for following, and bye.